We introduced smooth muscle when we talked about arterioles in the previous lecture and the important role that arterioles play with regulating blood flow. So let's take our time now to review the physiology of smooth muscle. So smooth muscle is given the name because it does not have sarcomeres like what we see with skeletal and cardiac muscle. Smooth muscle does have actin and myosin, but it does not have the regulatory proteins of troponin and tropomyosin. Because it's not organized into sarcomeres, there's another mechanism which anchors these filaments, and this structure is called a dense body, and a dense body is attached to the cell membrane, and this anchors these contractile elements so that when they shorten, when the contractile elements slide by each other, it will help change the size of the cell. So here we have our thick and thin filaments, and just as we saw in our skeletal and cardiac muscle, when we have contraction, those thick and thin filaments slide by each other. When they do, it causes a change in the cell size. So this is showing our myosin, our thick filament, and the actin being pulled by with the cross-bridge cycle. Now recall that these smooth muscles are typically sheets of smooth muscle, and they're lining the entire blood vessel. So when these sheets change shape like this, it's going to cause a change in the diameter of the blood vessel. There are two types of smooth muscle, and they're given the name based on how they're organized in our body. So the first type of organization of smooth muscle we call single unit smooth muscle. And this is smooth muscle that's typically present in the walls of blood vessels. So imagine that this is an arteriole. We have lots of these smooth muscle cells in the wall. This would be the lumen, or the hollow portion of the vessel. Now, as we look at these single unit, or these smooth muscle cells that are organized as this single unit, there's going to be some characteristics that we need to identify. First off, realize this is the most common type of smooth muscle, and it's present in hollow organs. We're focusing now on its presence in blood vessels, but it's also in hollow organs such as our GI tract. These muscle cells are interconnected with gap junctions, and that's indicated here by these connections between the cells. And as you recall, gap junctions allow the rapid transmission of electrical signals from one cell to its neighboring cell. We have pacemaker cells in single unit smooth muscle. So the pacemaker, as you recall, is a cell that can spontaneously depolarize. Now the benefit of having these gap junctions is if we have a single pacemaker cell depolarizing, that signal can then cause depolarization of neighboring cells, causing those cells to contract. Because these cells are so highly interconnected with gap junctions, they do not have to have a lot of neural input. So this relative paucity of neural input means that, yes, they can have neural input, but you'll notice that nerves only go to just a limited number of cells. But realize that if the nerve is what causes depolarization of one of these cells, that signal can then be quickly sent to neighboring cells because of the gap junctions. Single unit smooth muscle is unique in that if we stretch it, it will respond by contracting. So this is actual force that's created in the muscle in response to stretch. This is not just simply an elastic recoil. Single unit smooth muscle has a unique characteristic that it always seems to maintain some degree of resting tone or resting contraction. Now the other type of organization of smooth muscle is called multi-unit smooth muscle. And what we'll notice with this arrangement is that, one, it's present in different areas. So we see it present in some of the muscles around the eye, as well as muscles of our large airway. But you'll notice here that there's no interconnection between these individual cells. There are no gap junctions. So because there's no gap junctions, there's also not going to be any pacemaker cells. Without there being gap junctions, we also then are going to require a high degree of neural input because activating one cell with a neural stimulus won't do anything to neighboring cells, so we'll have a high degree of neural input, which each cell having a synapse. Stretching of multi-unit smooth muscle has no effect. They also do not retain any type of resting tone. So multi-unit smooth muscle, the cells act as individual cells. That's to be distinguished from single unit smooth muscle, where all of the cells act as a single unit. So this is showing our single unit smooth muscle and our multi-unit smooth muscle. And now the actual contraction in the individual muscle cells will be regulated by the amount of calcium that's present in the sarcoplasm of the smooth muscle cells. So that sarcoplasm term, this simply refers to the cytoplasm of these muscle cells. Now it's interesting that in smooth muscle, calcium concentration can be regulated by a variety of mechanisms. So one way that we can see calcium regulated as far as how much gets into the cell is that calcium may enter through channels. And these may be voltage-gated channels. So shown here on the left is an action potential coming along, opening up a voltage-gated calcium channel and letting calcium in. We may have a neurotransmitter or hormone that acts, say, via the G-protein link complex. And in this case, we're going to be creating IP3, which can then cause release of calcium from the sarcoplasmic reticulum, and that causes an increase in calcium. We may have a neurotransmitter or hormone work on a channel link protein, and that will open up the channel, allowing calcium in. As we mentioned with single unit smooth muscle, we may have a stretch gated channel that opens in response to stretch, letting calcium in. So the take-home message here is that there are numerous mechanisms that can cause an increase in calcium within the smooth muscle cell. And if we increase the calcium, that will lead to contraction. This is a diagram that we've borrowed from J. Perkins, and this is a nice diagram that begins to connect the calcium coming in to how we actually then will end up getting contraction. So regardless of the mechanism, we have an increase in calcium. In these smooth muscle cells, calcium will then bind to calmodulin, and this calcium-calmodulin complex will then activate this enzyme called myosin kinase, or in some textbooks it's referred to as myosin light chain kinase. That's just the specific area where the myosin is going to be phosphorylated. And that's what this myosin kinase does. It will phosphorylate myosin. So we can see that we are phosphorylating the light chain of myosin. That's the portion that's actually being phosphorylated. When myosin kinase phosphorylates myosin, we now say that this myosin is activated. So at this point, the myosin can bind to the actin, and the cross bridge will occur, and we will have sliding of the filaments by each other, and we will then have tension created. So as long as we have calcium present, that will activate myosin kinase, and we will have contraction occurring. Now there's another enzyme in smooth muscle that's called myosin phosphatase. So in reality, both of these enzymes are active. The issue of whether or not we get contraction or relaxation is which one is predominating. So when calcium is present, the myosin kinase is going to tend to be the one that has greater activity, and contraction will occur. As those calcium levels fall, then the myosin kinase activity slows down and the myosin phosphatase activity predominates, and that will cause the myosin to be deactivated and relaxation will occur. Now in smooth muscle, there's a unique configuration that we identify, and it is referred to as the latched state. And what the latch state points to is the fact that we can have actin forming a cross bridge with, or myosin forming a cross bridge with actin, causing the power stroke and holding that degree of tension. And that is called the latch state. So what that allows to happen is it allows these smooth muscle cells to maintain tension without using a lot of energy. So it's a very efficient way to maintain tension within these smooth muscle cells. Now I want you to know that the latch state is something that can occur in smooth muscle, but I don't want you to get lost in the details of how this might come about, because there seems to be some conflicting or various theories to suggest how the latch state occurs. As an aside, I will show you one of those theories, but this will not be something that I hold you responsible for. So I mentioned that in the smooth muscle, myosin kinase and myosin phosphatase are both present and have varying degrees of activity. One theory about the latch state is that when we have high levels of calcium, that both the kinase and the phosphatase have high levels of activity, and so we have this cross-bridge cycle continuing to occur at a high rate. Now, if the calcium level is only mildly elevated, then we'll see both of these enzymes active, but at a reduced rate. And at this reduced rate, we can have tension maintained without having a high rate of turnover, and that's when the latch state occurs. Now, a final term that we'll talk about with smooth muscle contraction is this thing called pharmacomechanical coupling. And what this identifies is a way that we can get tension within smooth muscle without having a change in membrane potential. Think back to skeletal muscle. In order to get contraction of skeletal muscle, we had to have a change in membrane potential. That change in membrane potential was an action potential, and that caused the opening of the activation of the DHP receptors, which opened up ryanodine receptors and allowed intracellular calcium levels to increase, and that caused contraction. So in skeletal muscle, we always saw a change in membrane potential preceding an increase in intracellular calcium. We see the same thing happening in cardiac muscle. But in smooth muscle, that's not necessarily the case. So on our top graph here, we show the membrane potential of a smooth muscle cell. And notice that our membrane potential stays at -50. But we're showing an increase in tension in that same muscle. Well, the way that that can occur is by a chemical messenger, and in this case we're showing that chemical messenger being epinephrine or norepinephrine. It utilizes a G protein link complex, which results in IP3, which causes an increase in calcium. So this calcium has been released in the sarcoplasm without any change in membrane potential. This calcium goes and binds to calmodulin, and that activates myosin light chain kinase, and that results in creation of tension. So the pharmaco refers to the fact that we can have a ligand causing tension without change in membrane potential. This is only seen in smooth muscle.